Hi students, once again I am meeting you to discuss about the applications of biotechnology, under which yesterday we were talking about, that is applications in medicine, under which we have studied how far the insulin hormone produced, then human black albumin, etc. Now the fifth one under this category, the human growth hormone. When insulin was made by different people using E. coli, another set of researchers concentrated on the production of growth hormone in the form of somatotropin and somatostatin. What is the difference between these two, somatotropin? Somatotropin induces a growth, promotes a growth, and the somatostatin inhibits the growth. It promotes the growth and this one inhibits the growth. These are the factors concerned with what is called the treatment of disease. Say an example of jaundice or dwarfism. Now, the growth hormone is secreted by the adenohypophysis, that is a pituitary gland anterior lobe. What is its function? It is concerned with the growth and development of the body. How? By enhancing or by increasing the uptake of amino acids, the one responsible for protein synthesis. So it increases the uptake of amino acids and promoting protein synthesis, thereby helping the growth of the body and the development of the body. Suppose if there is any deficiency, in such cases there occurs a condition what is called dwarfism, what I studied already in 11th standard. So a short nature of the body or midget. There you have retarded growth because of the deficiency of the growth hormone. So in such cases, now the dwarfism can be treated by administering or injecting the hormone secreted by or isolated from the pituitary gland cells. But we need large concentration of this hormone. For that we need the recombinant DNA technology. The same method what we used for, that is human insulin hormone. The same way we are just doing the process of recombinant DNA technology, producing the hormone, etc. I will represent it in the form of a picture. Okay? Through recombinant DNA technology, the growth hormone has been produced. Further, the gene for what is called human growth hormone is isolated from the human cell, particularly from the pituitary gland cells. Then it is normally cut, isolated by using the enzyme ECOR1, restriction endonuclease enzyme, which cuts actually the DNA at specific site, what is called the regulation site, resulting in sticky ends. So normally there are two types of cuttings. In some cases, some restriction endonuclease enzymes cut the DNA molecule with the sticky ends. In some other cases, they cut the DNA molecule with the blunt ends. Now we are using enzyme here, ECOR1 from E. coli bacterium, which cut the DNA, the gene, what is called the human growth hormone gene, with the sticky ends. Okay, this is number one. So we are getting the gene of interest, gene of non-function. Now the E. coli bacterium is taken, there is a bacterial cell, it's having, you know that one already seen in the insulin hormone production, bacterial DNA with the plasmid DNA. Now the plasmid DNA is used as a vector for transferring the gene for recombination process. It is being isolated, and by using the same ECOR1 enzyme, we have to cut open what is called this plasmid DNA. Now this is cut open plasmid DNA. We are having what is called cut open plasmid. Cut plasmid DNA. Using what is called the restriction endonuclease enzyme, the same ECOR1. Now it's cut open. Then we have what is called the DNA recombination. Now the gene with the sticky ends is made to recombine with this plasmid or inserted into the plasmid. That process called DNA recombination with the help of enzyme what's called DNA ligase. So restriction enzyme is used to cut open and DNA ligase is for joining. Actually it's a paste molecule. So the cut plasmid DNA is made to recombine with the gene of interest, what is called the gene for human growth hormone to form a recombinant DNA. Then the recombinant DNA that is normally incorporated into the bacterial cell. Now the bacterial cell E. coli contains the recombinant DNA. It starts synthesizing or producing or manufacturing what is called the growth hormone. Now the cells are separated from the colonies or from the culture, and they are being used for the mass production of the hormone by using what is called the fermentation technology. So if we want to get more number of molecules, we use what's called fermented time, as in the case of insulin hormone. The same mechanism being used here also to produce what we have large amount of or large molecules of human growth hormone by using what is called fermentation technology or using fermented time. Okay, so that's about what we have, the mechanism that is adapted, the same mechanism as in the case of insulin hormone production. Now, the recombinant form of hormone is called somatotropin. The recombinant form of hormone is called somatotropin. What is its importance? It is used as a drug for the treatment of diseases, particularly to treat growth disorder diseases. So the recombinant form of growth hormone is called somatotropin, which is used as a drug to just actually to treat growth disorders in children. Suppose the child is dwarf, then the hormone is administered or injected that promotes the growth, so the deficiency is rectified. Okay? Right. So that's about what we are the human growth hormone. We have to go for the next one, the recombinant vaccines. I mentioned about the use of growth hormone. The recombinant form of growth hormone is called somatotropin. That one is used as a drug, which is normally used to treat growth disorders in the case of children. So with that I concluded about what we have the growth hormone. The next one we have to pass on to recombinant vaccines. So recombinant DNA technology has been used in the production of new generation vaccines. Now, what is the advantage of this one? Mainly, it overcomes the disadvantages caused in the production of conventional methods of vaccine. So we produce vaccines by conventional method, the normal method, the traditional method. So the limitations of traditional vaccine, limitations means nothing but the disadvantages, anything else. So the limitations of traditional vaccine, that production could be overcome by this approach. What approach? The production of vaccines through recombinant DNA technology. Now, what are the main advantages of recombinant vaccines? The recombinant vaccines are, number one, generally of uniform quality. No differences, uniform quality. If you are producing one particular vaccine, it has uniform quality. And secondly, it has less side effects when compared to the vaccine produced by conventional methods or the traditional methods or the normal methods. These are the two advantages of what is called the recombinant vaccines. Now, what are the types of recombinant vaccines? Subunit recombinant vaccines, attenuated recombinant vaccines, then DNA vaccines. So these are the different types. Subunit recombinant vaccines. Number 2, attenuated recombinant vaccines. Number 3, DNA vaccines. Now we have to go for one by one about the role of each one, how far they are being useful and what are the components present in them. Okay, let's continue. What are the types of recombinant vaccines? There are 3 major types of recombinant vaccines. 1, subunit recombinant vaccines. Number 2, attenuated recombinant vaccines. Number 3, DNA vaccines. Now, what are subunit recombinant vaccines? The word subunit is given because not the entire organism is used, only the component part of the organism. The component part of the pathogen is used, hence called subunit. The meaning for that one, only the components of the organism, not the entire organism. So, vaccines that use the components of the pathogenic organism rather than the whole organism are called recombinant vaccines. Subunit recombinant vaccines. So only the units, subunits of the entire organism, not the whole organism is used. Now recombinant DNA technology is used for, actually is very much used for this development of subunit vaccines. New subunit vaccines has been produced because of what is called recombinant DNA technology. That's the best method. Now what is the advantage? Actually we are using only the units, subunits, and what are the subunits used for the preparation of that one before the advantage? The subunits, the components used normally for the preparation of the vaccine include 1. Peptides, 2. The proteins, and the DNAs of pathogenic organisms. I mentioned already, not the entire organism, only a component. Either the peptides or the proteins or the DNAs present in the organism, the pathogenic organism can be used. Now what are the advantages of this vaccine? Number 1, purity in preparation. It is very much pure nature. Number 2, stability. It is very stable, not undergoing changes. And number 3, safe in use. Doesn't have any side effects. That's why safe in use. So that's about subunit recombinant vaccines. And we have to go for the next one, that is attenuated vaccines. Now the second one, the type of vaccine, attenuated recombinant vaccines. That is, attenuated means the one which contains weakened germs, either in killed form or in live form. Now, this vaccine includes genetically modified pathogenic organisms. We have the pathogenic organisms, mainly bacteria and viruses, that are made actually non-pathogenic. Then they are used as vaccines. That's why I say normally this vaccine contains mainly the pathogenic organisms in modified form. So nowadays it is possible to engineer the pathogenic organisms and use as live vaccines. For example, measles vaccine, polio vaccine. These are all the live vaccines. So it is possible to engineer what we have, actually the organisms, and made into non-pathogenic and use as live vaccines. And such vaccines which contain either live or dead organisms together constitute what we have attenuated vaccine. If it is produced through recombinant DNA technology, that is called recombinant just DNA vaccine. Recombinant what is called attenuated vaccines or attenuated recombinant vaccines. So normally attenuated vaccines can be produced without recombinant DNA technology. If they are produced through what is called recombinant method or RNA technology or genetic engineering method, then it's called attenuated recombinant vaccines. Now the third type of vaccines what we have, actually DNA vaccines. Now it is used for mainly genetic immunization. So the genetic immunization of what is called actually a body, the genetic immunization of the body by using DNA molecule is a new method, a novel approach. A novel approach means a best approach, a best suitable approach. Normally that developed in the early, that is in 1990s. So this is about DNA vaccines. We have to proceed further about that one. So it is nothing but the genetic immunization of the body by using DNA. Normally the immune response is generated or produced by means of what is called DNA molecules rather than the entire organism or proteins, etc. Here DNA is acting as an antigen that stimulates the production of antibodies, thereby triggering the immune response. Normally the proteins or peptides or any other molecules are acting as an antigen, stimulating the immune response. But in the case of DNA vaccines, the immune response of the body is stimulated by the DNA itself. The DNA molecules acting as an antigen. They stimulate what is called the immune response. Now the DNA vaccine consists of a gene particularly, which is encoding for an antigen molecule. That antigen molecule is to be inserted on a plasmid. So that antigen is encoded by the gene that is in the DNA vaccine, is to be inserted on what we have the plasmid, then incorporated into the cells of target animals. Into which cells they have to be incorporated, in which animals. This normally stimulates, the DNA stimulates the production of antigen molecules which are being displaced on the surface of what we call the cells. Once the antigens are produced, these evoke or provoke what we have the antibody molecules to work against the free floating antigen secreted by the cells. This is the mechanism. So the DNA is inserted, that is a G, to a plasmid and incorporated into the cells. Now the DNA instructs the cells to make up or to produce or to synthesize antigen molecules. These antigen molecules are displayed on the cell surface. Then this one provokes or evokes what is called the antibody molecules to fight against the free floating antigen molecules secreted by what we have the cells. Now, normally these DNA vaccines cannot cause diseases. The reason for that is because it contains only copy of a few of its genes. That is number one. Another advantage also we have. So it is relatively less expensive. It is relatively easy to design and produce vaccines. These are the two main advantages. In addition, we have some more advantages. We will see that one. What are the advantages of DNA vaccine? The DNA vaccines cannot cause disease because they contain only a copy of a few of its genes. A copy of the genes only, not the original one. A copy of a few of its genes. Maybe it is the original gene, but only a copy of a few of its genes. Not many genes are present in the DNA molecule, which can be used as a DNA vaccine. And that is one of the advantages. The second one, the DNA vaccines are relatively easy and less expensive to design and produce. That's about the advantage. Now, what are the common advantages because of the production of vaccines through this technique, what is called recombinant DNA technology? Number one, producing target proteins. Only the target proteins. What protein we need, that alone being produced by recombinant DNA technology. Number two, long-lasting immunity. Once immunity is given by these vaccines, it is lasting forever. Number three, it triggers immune response only against specific pathogens with less toxic effects, not attacking all the pathogens. You see that one? The immune response is triggered by these actually recombinant vaccines only against specific pathogens with less or least toxic effects. These are all the common advantages of vaccines produced by the new techniques, what is called recombinant DNA technology. These are the advantages of DNA vaccines. They cannot cause a disease. Number two, it is very easy and less expensive to design and produce a vaccine. That is about the advantage of DNA vaccine. This is the advantage of all the vaccines produced through recombinant DNA technology. Okay, that's about this one. We have just next one, the last one, edible vaccines. Edible vaccines. The edible vaccines are prepared by molecular forming method. What is molecular forming? The production of therapeutic chemicals and drugs. That is what we call the medicines. By using rDNA technology is called what is known as molecular forming. So, and by using the genetic engineering technique, what I mentioned inside is nothing but actually genetic engineering technique. So the production of what is called therapeutic chemicals and drugs by using rDNA technology or genetic engineering is called what is known as actually molecular forming. So edible vaccines are prepared by this method with the help of rDNA technology. Now, what happens here? The selected genes are introduced into the plant cells so that we can produce transgenic plants. So the transgenic plants once formed which manufacture the encoded protein, the gene encoded protein synthesized. Normally, the edible vaccines are specific in their actions. The edible vaccines are mucosal targeted. Mucosal targeted means attacking the pathogens found in the mucosal layer and cause stimulation of both systemic. Systemic means the whole body where you have the mucosal lining is present. So both the systemic and mucosal immune response. Whenever pathogens, any organisms found in the whole body, particularly in the mucosa, and we have the edible vaccine can cause. Because the mucosal layer is present, the respiratory tract, the digestive tract, urinary tract, etc., where we have the accumulation of pathogens sometimes. So the infection of the mucosal layer has been removed, has been cancelled, has been treated with the help of what's called edible vaccines. That's why I mentioned edible vaccines are mucosal targeted, targeting the pathogens found in the mucosa. Now, it is used for the treatment of a variety of diseases, both animals and plants. For example, in human beings, the measles, then we have hepatitis, and also cholera. And in the case of cattle, we have foot and mouth disease. It is one of the diseases treated with the help of edible vaccines or subunit vaccines. So foot and mouth disease, a disease in cattle, and other diseases, measles, hepatitis and cholera are the diseases of humans, all being treated with the help of edible vaccines. Okay, the last part is actually how far you see that one, the recombinant vaccine for hepatitis B synthesized. The mechanism will be the same as in the case of human insulin hormone. Okay, let us discuss.